sound suddenly disappeared. Can you hear me?
Yes.
Hi.
Hi.
I can't hear Susan. She was talking to me.
Oh.
Okay. Can you hear me now?
Yeah, I can hear you now.
Okay, you can hear me now? Okay.
Yeah, yeah.
So the wild species...
Susan, can you hear?
They've dug out.
Oh, my God.
Pardon, can you hear me?
That's okay.
Yes. I can hear you.
You can hear me?
Yes.
Right.
Oh, there we go. Okay.
Yeah.
All right. Yeah. Anyway, so there's a dog.
Okay.
Who dug up wild plants.
Bradley, you might want to contact Manic Deb.
Okay.
He's got exams this week, but sometime in the near future, he wants to do a presentation on our work on polygonal shapes of archaea. And he wants to, we're looking for a critical review of whether we've covered everything, because we've got over 300 species to go through, and we don't want to have to revise what we're doing in the middle.
Oh, okay, yeah.
Okay.
Yeah.
Okay, so we need a real critical statement. Have we covered everything? Have we used the right algorithms, etc.? Okay?
Yeah, that sounds good.
Okay, so why don't you schedule that with Mike?
Oh, yeah, I will. Yeah.
Okay?
Yeah. No problem. Sounds great. I'm glad he's making progress on that.
Yeah, he is. He's doing beautiful work. It's just that I don't, there are all sorts of methods available. And whether he's using the best for what we're trying to do is sort of the question. Okay?
Okay, yeah.
Okay?
Yeah.
And it's tough because there's all sorts of thresholds involved. The 300 pictures are taken under 300 different conditions.
Right.
Okay. We have to include quality measures of the images, things like that, because obviously the images of higher quality will presumably produce more reliable data.
Right.
Okay. So there's a lot of questions as to whether or not all the methods he's gathered for doing this are the best.
Okay.
Okay.
Yeah, that sounds great.
So I'm going to shut off my video.
He's doing a hell of a job. It looks beautiful.
Oh, that's good, yeah. I knew he was showing a little bit of it before. He had like a early preview of it, several months ago. So.
Yeah, we're ready. We're trying to get ready to do the mass production.
Okay.
Going through 300 images. So, as I said, we don't want to have to do it twice.
Yeah. Where are you getting the images from?
Most of them are from microbiology society journals, but they're all over the literature.
Okay.
Okay. And they're pulled off of various journals. A lot of them are screenshots, so the quality issues are important.
Right.
Okay, because I can't get the originals.
Yeah. Yeah, but it should be enough to do a good analysis. I mean. Yeah.
The question, yeah.
Good.
See, some of these things are weird. People will show a picture of these archaea and without any comment, and they won't notice that sitting right there in the middle in the picture is a beautiful triangular cell.
Oh, right.
They don't even mention it sometimes. It's as if they took the picture just to take the picture and didn't bother looking at it.
Yeah.
So we've got lots of those, and so his methods will pick those out. Okay.
Yeah.
And quantify.
Do they at least have this size scale on them? Your pictures? You don't know.
Okay, that's all I've got for today.
Oh, Susan asked if it had like a scale bar on the images. Probably not, because they're just.
Usually they do.
Yeah. Okay.
Yeah, in fact, he has to remove the scale bars so they don't mess up the image, the analysis.
Right. That sounds great.
Okay.
All right. So it looks like Karan's here. I sent Karan some data from a couple pieces of data from Zebrafish. And I don't know if you've been able to take a look at that, Karan, or...
Yeah, hi. I'll just. Would it be okay if I share my screen and show whatever
yeah, go ahead.
that are there?
Yeah.
Even the axolotl data that I wanted to show you. Because the thing is with that axolotl data, wait, I'll just share my screen first and I'll explain.
Okay.
Uh,
You know where the screen share button is? Or?
Yep.
Okay.
Yeah. Is it
There we-
visible?
Yeah,
Yeah.
there we go.
Okay, so instead of going through the whole database, I just selected I think one set of images that I think are repeated for different samples. If I'm not wrong, there were like 9 images per sample, if I'm not wrong, because then I saw this pattern that was recurring here. So far for visualizing an image of this sort, in like had there been more metadata related to this image, I think it would have been a bit more easier, like let's say angles and things like the depth of this thing that is there, and the at least some scales with respect to the x, y, and z axis.
Yeah.
So because those kind of details are not mentioned so far, so what I've done right now is I'm trying to I've plotted contours of first I've done some pre-processing here, like I've scaled these images to a particular size, to make it a bit more reliable in the process, a bit more easier.
They are
for
approximately 2 millimeters in diameter.
Okay. 2 millimeters, okay. Okay.
And I'll get you better data. I just need some salamanders to lay some eggs. And then I'll be able to give you angles.
Okay. Because otherwise, what I was thinking was, the plot here shows, at least gives some depth related to the background that is there, the RGB values that are there. With respect to those values, I think I was, trying to, I was thinking of, plotting that 3D data in some way, like in that Zebrafish data also, data set, like these are the XYZ values that are there. So the thing is, this SSBD database, even though it has, things that are there, like every file is associated with this BDML ID that is there,
Yeah.
which stands for, I think, biological data something. So yeah, the thing is, their BDML IDs for some data sets is not exactly functioning properly. Like if I go to download the BDML file directly here, it shows that resource is not there. So I had to use their API for this. So this API thing, again, they've kind of mentioned, how to go about using it. So this is the kind of raw data that it gives out for arranging and viewing it and then displaying it as an entity here. I was trying to go along these lines, but this so far is in position of a nucleus is present in that, if I'm not. So even these files are not there, I think. So this thing, I think, would take, which I still have to work on,
Right.
finding out what these T values here mean exactly. Because if I'm not wrong, the T stands for time scale, and XYZ values display the movement of that nucleus or some more IDs that are there. This is the object ID that's there for there are like at least 63 IDs per time scale value.
Yeah.
They keep on decreasing. So
Yeah, the ID is a cell, like a cell number. So it's, like for each time point, there are a certain number of cells in the image. And then, it's like 1 through 63, and they just have the numbers. They don't really have names for the cells. But then if you plot them out by an XYZ value, you get a plot, like a 3-dimensional plot. It kind of looks like the embryo would look. And, it's just kind of this point cloud. That's what it should look like. That's what you should be able to get out of those data.
Okay.
Yeah.
Okay, so I was trying to like for the axolotl data set. As of now, I'm thinking something along those similar lines. Like naming these as objects and and plotting them according to the pixel value, and associating that pixel value with the x, y, and z axis so far. But if there's more data, then I think things will be slightly more easier. I think, like with respect to angles and the XYZ scale, like 2 mm diameter if it's there for this. So I think it should give something, at least something better than this current model right now.
So I mean, yeah, this is good.
Still, there's a lot of like tweaking around that I'll have to do before coming to it. But yeah.
This looks very good. I'm really pleased you're working on this. And these pictures were taken kind of at a random angle, so what you've got there, I'm really happy to see this.
Okay.
Yeah, I just need to get for it. And unfortunately, that's
also regarding
some animals that my friend has, who might lay some eggs for me.
Okay.
Yeah. So that one of the projects we were going to do with the axolotl was to create like an atlas where we took each one and just kind of rotated the embryo. But we also, we need to sort of analyze the data itself, the cells within the embryo and that. So I like this approach where you're just kind of like going at the images and trying to put them into some context. It's good.
Yeah. Some depth layers,
Right.
Like I could maybe stack them with different slices, something like that, maybe.
Yeah.
And also regarding that, sideline idea with respect to that viscoelasticity in tissues, right?
Mm-hmm.
So the stress models for that again was something that I was trying to,
you cut out there.
Like, they're like, is it not visible?
No.
Okay.
Okay, your screen's back. You're sharing.
Okay. Yeah. So I think like at the end of this paper, I found some methods of finding data sets for this particular reason. Because when it comes to I think creating a stress related tensor model for any medium, if it's a physical medium like let's say a piece of metal or all, then it's okay. Then it's kind of easy. But when it comes to tissues, I think there's a lot of like continuum equations involved and all that. So just the image, I think, won't do for that case. Otherwise, that is still something that I need to look into a lot. That I just, because there was this again an idea, like we were doing, powering implantable medical devices. So the stress model of something like a tissue would kind of give more perspective into how we can have some more powering methods for implantable medical devices, because a lot of them have these piezo electric cords. So if we have like for various sites in tissues, maybe in the human body or any data set that is available, I'll have to again look for tissue-based data sets for stress models. So that could kind of help out in that. Just additional thought towards that viscoelasticity in tissues paper that wasn't mentioned in the last lecture.
Okay, two things. One, this is, I'll hold it up. I don't know if you can see that. Not very well.
Yeah.
It's the microscope that I'm going to use to take images of the eggs, and you can see multiple microscopes at different angles.
Yeah.
So it's a 2 millimeter egg, and that's the microscope. And the other, can you give us a reference for the paper you just showed us?
Yeah, just this was, I think, Mm, yeah, this is various models for modeling stress and strain equations of in tissues. This paper is, I think, I'll share the link here.
Okay, there it is.
Okay.
Mm, yeah.
There it is. And did kind of
Yeah,
question
this
here in
is
the
this
chat.
is another summary of that. Yeah,
Yeah. Oh yeah, that looks
like
nice.
these are the techniques that are there for. I think that similar thing. This is a different paper, I think.
Oh,
Mm,
okay.
yeah.
Oh,
Uh,
that's. Did you
this
pick the
is
link to that too?
yeah. This
Can you
also.
go down the page again? That was what, of living cells?
This was again these are different techniques used for modeling that same stress related tensor models or anything for anything related to finding stress and strain. Uh, what do you say? It's like various ways of gathering data. Like 2D data is there, 3D data is there in different conditions like in vitro, ex vivo, and the methods that are there to calculate stress for those tissues using these uh,
Um,
methods.
could you show the front page of that
Title,
paper?
yeah, yeah.
Because then I just do a print screen and then I've got it.
This,
I can look it up. Okay, so that's
yeah,
what
yeah.
that's called, measuring.
Measuring mechanical stress in living.
Oh, okay.
Yeah,
I'm pretty sure
I'll
I
post
may
this link
have
also.
that one already,
Okay.
but
Yeah, yeah. Because this is kind of like first ones that come forward when you start going through similar kind of papers. This is, I think, was published in 2009. Yeah.
Yeah.
and have you just showed us the two papers?
Yeah, like these are the only two papers that had some leads, that I need to go through for creating that 3D tensor model. Like for at
Oh,
least some
okay.
ideas, because the thing is, Python libraries have very little support for modeling issues like this, so I'll have to find some third-party software, along with this to maybe get some more data. But then again, this will take some more time, I think, because there are a lot of things to go through before I even start getting some proper model out of it. So this
Okay.
was there.
Yeah, I'm working on this myself. Like the title of that is exactly the topic I'm working on right now.
Okay. Okay. So
So,
like,
oh,
uh,
go ahead.
yeah.
I'll
Yeah,
turn off.
so these three things are, kind of there side by side. And see, the thing is, with the Zebrafish data set, I think again I'll be getting like positions of respective nuclei, with respect to each other, and I'll be able to model that and all. But with respect to the axolotl thing, yeah, I think you using a different way of modeling this, I think, would be more practical. I still have to, I think, go through more things. But yeah, these are the three things that are currently
All right.
there that need more work. Yeah. Paste the other link also.
Oh, stage one. Okay. I was going to say, Dick had a question about what stage were the axolotl embryos, and Susan said they look to be stage one. So,
Okay, that explains
These,
why you can't see cell movies.
uh, these are the first 9 images for the first sample, I think,
yeah.
that I'm trying to model.
So the way that these are captured is that they actually are flipped in a like in a water column. So the cells are flipped from bottom to top or something like that. Is that correct, Susan? Or
Yes, and it's just one cell. That's one cell.
yeah, so that's the one cell state, or it's a single cell, and you're just looking at the rotation. So you're able to get like the surface along most of the surface, I guess, of the sphere. I don't know if there's any missing area.
There is. There would be missing area because it's just flipped in one rotation instead of, and the sideways rotations are not there.
Okay, because I think these images were kind of similar, so I was thinking of maybe clubbing 3 or 4 images like these, which look similar, and then plotting like from one perspective, like these 3 from the top view, these 3 from the bottom view, something like that. Again, with the pixel value that's there.
I should use this new microscope, like I said, and get you images with definite angles. Or it'll be sort of approximate.
That would definitely help you.
But definite directions.
Yeah, then it'll be a lot more better, I think, because this is just the 2D slice. Right? Yeah, that would definitely help me out. Yeah. This is then it's mostly, I think I was trying out there are 2-3 libraries that are there. Naparis is there, ski images there, like for plotting 3D models and all that. So this thing is still going on. I need to work more the X, Y, and Z axis.
So you're using OpenCV or Scikit Image, or using both of them?
Image processing part,
yeah. And so you're just like playing with some algorithms, trying to figure out what the best ones are.
What would fit the model best? Yeah,
we've had people do this a lot in the group. Trying to work with embryo images and trying to figure out the best algorithms. So, it really depends on the kind of microscopy image that you have. It depends on if you have good edges, or sometimes you'll need to do some pre-processing or augmentation of the data set to get really good results.
Yeah.
So, those are things that we've played around with. And with embryos, it's kind of hard. You really have to focus on, how the image is separating from the background. That's one of the main things that we found.
There were 2-3 things, for pre-processing.
Yeah.
Something similar only, like resizing was done, grayscaling was done,
Okay.
Because these images are, I think, 2992, like 3000 into 2000 pixels, something like that.
Okay.
So I, scale them to like 1/10 of their size for the plot to, show, like exclude some details that were not important. But I'll have to again tweak these to see, depending on the new images,
Right.
But this was most of what I've done along with pencil. I'll try out more things by the next week.
Well, that's good. Yeah, keep up the progress if when you can on that. And a couple words on the Zebrafish data set. So that whole data set is, I can't remember the extent of it. I sent you that other link to the ZFin database, which basically has like all the stages.
There were two links. Yeah.
So
like these IDs are there, and I had compiled there because these resources were not available. I had to access their JSON files. So they have this weird schema, of getting their data using their API. So I'm still trying to work around it because they're like 63 objects, per time scale.
Right.
Like this is time scale 1, and there's like this is 1. So getting all that together directly to the Colab notebook, that is what I'm trying right now so far with this Zebrafish object thing. Otherwise, the thing is their website does show the 3D image, like it shows here how much time it will take to render that 3D data set and how it will change in time. But the thing is, after it reaches 100%, it just stops there. It doesn't show the variation in the data set with respect to time. So even though it's mentioned here X, Y, Z, and T, So I think that part has some issues because again, some resources that their site is supposed to have is not there.
Right.
So far, I've at least downloaded the JSON files here. So I'll work through them, and then I think it'll be at least I'll be able to build something based on that.
Oh yeah, no problem. And well, just so you know that like the 63 is just the cells at that one time point. So like as you
Yeah,
move
yeah.
towards
Then they're
different
like
time
more
points,
and
you're
less.
getting
Yeah.
more cells, but you won't know the ID of. You can't go from like you can't track any one cell through time necessarily. It's just going to be like 1 through 63 and then 1 through 74, and then it's going to grow, and then you'll see definitely that you're getting more cells, but it's kind of hard to get
Okay.
like to track any one cell. I mean, you can look at the cells from time point to time point and look at the sort of the aggregate trends, but other than that, it's going to be there's no identity that you can track a cell and say this is
Okay.
yeah.
Okay. Okay. Otherwise, I was thinking, maybe there'll be some because if the cell divides, maybe we could have some sort of a family tree going, maybe. But at least the cell populations will be another parameter, that
Right.
maybe.
Yeah.
Yeah.
It's in C. elegans you have the cells have a distinct name, and that's because of their mode of development. They're deterministic, so you can trace lineages back, to a common
Okay.
founder cell, but then of course you can do
Okay.
that in zebrafish, but it's much harder because you'd have to have like you'd have to use like a fluorescent tracking dye or something in the cell to see how it's passed down, and that's like a separate set of experiments, but that's
Okay.
right.
That sounds more
Yeah, it's a
complicated. But
different way
yeah.
to do that.
Okay.
Yeah. So thank you for that update. That was very good. Um,
Okay.
yeah. So why don't I go on to some of the other things that we had? I can share my screen here. Yeah, there we go. And we have a couple updates here. First thing to note is that there's a conference coming up, Dynamics Days 2022. And this is something that's been held every year. This is a nonlinear and dynamic chaos and nonlinear dynamics conference, and it's usually held different universities around North America. They have a European version, which is separate from this. I think I did the European version this summer, so they have one. But this is the North American version, and this year it's being hosted at Georgia Tech, but it's being held virtually because of they still hold them virtually. Last year they didn't have one at all. And then the year before, I can't remember where it was. They move around to different universities. But this is a nice conference to attend. I don't know what the registration, usually it's not very much, but the registration fee. But they always have some really good talks, and they always have some really good mix of topics from hardcore chaos, which is like looking at systems that have chaotic behavior, to nonlinear dynamics in biology and social systems in geophysical systems. It's a pretty broad range of things. And this, I've put in an abstract for this conference, so you might want to attend. And they have a lot of good speakers. They have people from Randy Beer, who does a lot of agent modeling. He's doing a lot of minimal cognition and things like that. Brad Ermentrout is a brain scientist. They have some other people here. Elizabeth Munch is a topological data analysis person. So it's going to be a nice set of talks. And this is held the January 7th through January 8th. So it's like right at the beginning of the year. It's kind of like in the still in the holiday break period for people. The term hasn't really started yet. And I've been to the one in Evanston, Illinois, which is Chicago, in January. And it's not typically a time you want to visit Chicago, but it was a pretty good time. So here's a link to the site for the conference if you're interested in attending. This is virtual again, so check it out if you're interested. I've submitted this abstract. This is, I think I did this one for Dynamics Days Europe. So this was last summer. And so I'm just kind of recycling this abstract and maybe making some changes. So this is this game theory of developmental processes talk that I gave at Dynamics Days Europe. And I have the video on the YouTube channel for DevoWorm. So this just kind of walks through using game theory as a tool to look at developmental processes. So we're looking at things like the shape of eggs or pattern formation or these different types of things where agents are trying to solve a maze. And you have this idea of developmental agents, and this is a developmental agent developing, and then there's an ontogenetic agent, and there's coordination, there are trade-offs. And it kind of goes through a lot of things here that we're interested in, what you can do with developmental game theory. You can, have a number of different types of games, like zero-player games, which is like this Conway's Game of Life. You have the payoff matrix. You can have one-player games, like Games Against Nature. You can have two-player games, like tic-tac-toe or noughts and crosses. And that maps to a dividing embryo, and kind of going through that. And then some other sort of behavioral evolution or behavioral development with observers and emitters of certain behaviors. So and then kind of get into developmental stable states, which is a idea that builds off of evolutionary stable states, and then a route to understanding combinatorial complexity of development. So again, this is on the YouTube channel, this video from Dynamics Days Europe, and you can check it out if you're interested.
Bradley, can you send me a copy of that?
Oh, yeah, no problem. I will.
Okay, thanks.
Second one is that I know we've talked about this for a long time, but I finally got a chance to get into this a bit more. And this is a DevoLearn preprint that we wanted to put together. So this is Mainak, of course, and Mayuk, and Ujwal, and myself. And this is the work that we've done on DevoLearn over the past 2 years. So we've done a lot of work on the software, done a lot of work on sort of the educational aspect, the web presence, and then promoting it at different talks. And then now we have this preprint. So it's a short abstract, a summary of the platform. This is in Markdown, so this is interactive. You have this GIF of this is something I think Mainak made of this sort of his, this is him using the interface. So this is a Heroku app that he built on top of the software. And so he's able to show kind of how it works. So you go into the interface, you click what your selection of what you want to do. You can predict lineages, you can do this sort of thing. And then it shows you the interface here. You can do some interactive analysis in it. I've been kind of putting in different parts like accessibility, then there's a statement of need, and I have the technical details. I need to add a couple more things in here. And the formatting needs to be worked out a little bit better. But I'm coming along on this, so I just wanted to give an update on that. It's a lot better than it was. And so I don't know where we're going to submit this. We'll have it as, at the very least, I want to make sure that we have it in place for the GSOC period that's coming up at in mid-January or late January. I think we need to submit projects for GSOC. So this is going to be like, something that people can see and people, oh yeah, this is something that they did over 2 summers worth of work. So, yeah, like I said, I might add some things on the lineage prediction, which I didn't do, but I'll just write that down. All right. And maybe some other things, but I wanted to get that I want to get this going, and I don't exactly know how we're going to put this together because we have a lot of GIFs in it. I don't want to publish it to a. Well, I'll probably have like a static version, but I like these GIFs in here, so we'll see what we do here with this. So the next thing I wanted to talk about, actually I'll talk about GSOC in a minute, but I wanted to talk about this. So this week is the Neurops Conference, and for those of you interested in machine learning and deep learning, this is sort of one of the hallmark conferences of the field. And this is, again, it costs something to attend. You can still register, but they're going to have a lot of the videos, I think, on YouTube eventually. So if you miss anything, you'll be able to see it there. They're also having, I think, this week and next week, they're having workshops. And so, let's see if I can find the workshops. This is the online proceedings, I think. So this is the online proceedings here. So this has all the papers, actually. A lot of these are put on bioarchive, so if you'll be able to access them. But there's just a lot of stuff in this conference, and it's very hard to kind of, you get hit with a lot of things. It's like the fire hose approach to this sort of stuff. So you want to figure out what you want to look at. So, I don't know how they have this organized. You might want to look at the proceedings, and they don't have a lot of, like, applied work. So the stuff that has to do with analyzing microscopy data, I don't even know if they're doing stuff like that in the main conference. But you might want to look through some of the papers to see what they're doing in there. And I was looking for the workshops. Oh, here they are. Okay, so there's the workshops, and these again are these are closed for now, but you might be able to get access to some of the videos on YouTube or on the websites. I think they have links to some of the websites here. So there's some of these workshops are very specialized. I don't think they have anything for biology again, but they have things like if you're interested in federated learning or metacognition in AI or databases in AI, which is closer to kind of what we're doing here. Machine learning and structural biology. OpenWorm had a workshop here in 2017 on, it was called WNIP, or Worm Neural Information Processing. And so they had a bunch of papers on, how, like a lot of papers on simulating biology and computational neuroscience, that sort of thing. So, there's some interesting things buried in here if you want to look through and see if you can find something that's interesting to you. But there's a lot of stuff here. Deep reinforcement learning. So that's that. Then I'm going to talk about Google Summer of Code, which is coming up. Like I said, the application period starts in the spring, but we have to have the proposals out by, I think, the middle or end of January. So this year, I think I mentioned it a couple weeks ago, it's a much more flexible program this year. They're going to do, short projects or long projects. We can do different things depending on what people want to sponsor. We can do, a very short project or a longer project. And then we submit it to the INCF, which is our sponsoring organization. Then they give us some slots. And the reason I bring that up is because every year we have a couple of projects, and then we have one project that gets selected usually. So we want to make sure that we have a range of projects, and so we can see who applies to what. And then, we don't really know which project's going to be accepted ahead of time, but we want to make sure that we give people an opportunity to get a taste of what we're doing, maybe, start some work of their own if they don't get selected. If they get selected, great. But it's pretty competitive. So this is something that's going to be coming up soon. And so we have to start thinking about it now, what we want to do for it. Again, we have something related to the DevoLearn project, or DevoLearn platform. So, that might mean improving upon DevoLearn, although I think for now, I think we've done a lot of work on the main software, and I don't know what the next step is. Maybe it's expanding it from like C. elegans to another species, or maybe it's working on some other aspect. We still have the platform that exists. So, there's the C. elegans the pre-trained model that we have, and then we have another couple of machine learning platforms that are more general that will analyze different types of embryo data. Then we're working on this stuff with diatoms. So these are these little single cell or colonial organisms that move around, and they have these very simple morphologies compared to C. elegans. And so we're working on techniques for analyzing that. We also have the axolotl, which is the working with an image or an embryo that's moving around. It's flipping, so we can have that sort of information about its surface and about its movement along this, rotational movement. So we have a lot of different things we can focus on. So I'm going to work on thinking about some of maybe the potential projects, and then I'll pull it together. Maybe our first meeting of next year, maybe I'll go over some of these projects, because I'm planning on maybe having one more meeting this year for DevoWorm, and then in the new year we'll start over, and we'll get
My new data won't be flipped. It'll be stationary.
Okay.
And I'll sequentially take pictures from all the angles that I've got, which are 9 or 10 angles, and then I'll leave the embryo for say 15 minutes, and then take another set of 9 or 10 images. Like, that's what I was planning to do.
Okay. Well,
as long as I can stay up.
Yeah. Well, that's fine. Yeah. I mean, I don't know how we want to do like an axolotl project. Last year, I know we suggested that people, just kind of model it as a 3D sphere and then put the data on top of that. And that, we've had people attempt it, but, we didn't get a finished product. And of course it didn't get funded. So there's that. But I'm just trying to think if, how we could make that stronger. And this might be it, having a different data set that we could use as a reference, or, just basically coming up with something that will analyze something like an axolotl embryo, which is much different than a C. elegans embryo. And I think, from the pre-trained model work, that the pre-trained model is going to be very specific to a certain species or a certain type of data. It's going to be limited, and will allow you to really kind of just identify stuff pretty accurately, but it's going to be very limited in it where it can, what it can analyze.
Limited. If you could make programs that did each species, that would be fine. I mean, each species has different ways of developing. One salamander from another salamander, they do it differently. So, I mean, in a way, that's okay. It's just, I guess, more work to come up with the product. But they are different species. They develop differently. They look different. Yeah.
Yeah. That's, there's also, I don't know, I usually have students kind of propose different techniques, what they feel comfortable with, and I leave that open. So there may be some emerging techniques that we might be able to leverage that would allow you to do something with, different species. I've been interested in this because I'm like, well, it's great that we have something that we can work with one species, but, development's pretty diverse in the sense that you have all these different types of embryogenesis and embryos, at least, in terms of from the standpoint of the algorithm. It's not doing exactly the same thing for every embryo. It's got to find cells, and the cells are dividing, and then, it may be picking up on something in C. elegans that's very specific to C. elegans, like the cell lineage, the organization of it. But then, in another species, that's not going to work at all because it's not organized in the same way. So that's going to be, you know, a problem, and I don't even know if there's a general solution to that, but it's, you know, something that we have to kind of find maybe the right data set to train the models on, or to figure out like what exactly the algorithms are picking up on.
Okay, I saw an algorithm on point clouds recently. Do you know anything about that, or?
I'm not sure. What was it? What were they doing? Like, was it biological?
Yeah, imaging things, microscopic images, I believe, with the point cloud. I'll try to find that paper. I've been dealing with too many papers. I've got like 5 binders full of the most important ones, so that I can sit and read them.
Oh.
And anyway,
yeah, that's always tough. It's but yeah, that's good. Yeah, if you could send that along.
I'll try to.
Yeah.
Oh, good. I want to show some pictures of diatoms with some mysteries about them.
Okay.
Okay.
Let me unshare my screen here. There we go.
Okay. Right on screen.
Why is it not? Let's see. There we go.
Okay. Share screen.
Yeah,
Okay.
share screen. All right.
Share. Okay. Still can't see it.
I can see your screen. Yeah.
Oh, you can?
Yeah.
Okay, let me show you.
Change your tab. Yeah.
Oops. You see the diatom?
Not yet. You're not on the same tab. You're on the Jitsi tab. You need to be on. There we go. I see
Oh.
it. There we go.
Wait. You see it now?
Yes.
Okay. This one here really mystifies me. This is a diatom which has lines of silica that are parallel to groups. Okay?
Yeah.
The question is, what on earth is going on here? What rules does this follow?
Right.
Okay? Okay, that's one problem. I've only seen one picture of this. Okay?
Yeah.
Another one had broken rapes, like this. Okay, the part which is motile doesn't go across the whole cell. It's broken, and the patterns radiate from this shape. And I have about, I don't know, I guess around 90 electron micrographs of cells, each one different. Okay, so it might be fun to see if one could isolate the shape of the rape and produce the pattern from it.
Yeah, that would be great.
Okay,
Yeah.
so there's 2 problems.
Yeah.
With 2 unsolved problems with that.
Yeah, they look like seashells in a while. Well, yeah, they do
Yeah,
actually.
it's a single cell. Yeah, let's see. Here's one that's got this is more typical, nice branching pattern. Okay,
Right.
but this one which has parallel lines has been quite mysterious.
Yeah.
Okay.
Okay. Yeah.
Okay.
Okay.
So anyway, let's throw that out. That's in my old 1994 paper on the chemical basis for diatom morphogenesis.
Okay. Yeah.
They've been sitting unsolved since then.
Oh, okay.
Okay.
Yeah. That sounds like something that we could, well, like a cellular automata of some type might be useful. And maybe one of the neural cellular automata that we've been talking about recently in meetings. So I don't know. I know that Shruti was interested in that at, I don't know if she's still interested in doing that, but.
Yeah, we worked on the diatom motility together.
Yeah.
She hasn't expressed any. We're working on detecting and destroying breast cancer right now.
Okay.
How about the diatoms in space before we get off diatoms?
Yeah.
Do you want me to revive that in a meeting at some point, Dick?
Well, what do you have? It's new. I mean, we have the proposal to send diatoms in space. We have the failed efforts. There was a high school group that actually put some diatoms in space, but the machinery for adding water to them once they got up there didn't work.
Oh.
So they went up dry and they came down dry. And I don't think it's been tried since then. That was many years ago.
Yeah.
I still think it's an important study.
It's typical of space studies. I remember from the 1960s, there was some piece of equipment that was guaranteed to work 70 times, so they tested it 150. And when they launched it, of course, it didn't work.
They launched it at 10 G's. It's a wonder anything works.
Yeah. So anyway, so diatoms in space. What, Susan, do you have something besides that?
No, I could look at it again, see if there's something to add.
I mean, we have Fred Hoyle's silly proposal that diatoms come from space. But
well, it's just that the way they grow in space might give you a clue as to what's happening to cytoskeleton in general.
oh yeah, I agree. I mean, there is work on
Very
microtubules.
important.
Yeah, there is work on microtubules in space, and they do. If you make a solution of microtubules, they do organize differently in space.
Ah. Mm-hmm. And our poor cells have to work harder to get them to go in the right direction.
Well, maybe.
Well, I think so. I think it's an energy problem.
Well, maybe. You think that all the astronauts might come back brain deranged.
When they come back with difficulties. I have a paper on it. Yeah.
Yeah, the brain is full of microtubules.
Yeah.
Hey, maybe we should launch nematodes in space.
I think, haven't they done that? I thought they had done that.
They may have.
Yeah. Sounds like something
Both
that's,
they have.
yeah.
Do they work?
Well, I think so. They're pretty hardy. They've recovered nematodes from like 30 or 40,000 year peat from like the
Oh yeah,
tundra.
that's that's in gravity.
Yeah.
Yeah, send all your model animals into space and try to figure out what's going on.
Hey, the archaea have the record. They've been recovered at 400 million years old from salt crystals.
Wow.
Yeah.
Salt crystals?
Yeah. Yeah. So actually, inclusions. They're inside water inclusions inside salt crystals, and you can actually get viable cells out of them.
Wow.
Do you have a reference?
Send me a note and I'll send you some. Sure.
All right. Well, thanks for that. Can I share my screen now, or?
Yeah, I've got a meeting in a couple of minutes.
Okay. Yeah.
Okay.
All right. Sounds good. So let's see. Let me see if I can share my screen here. So it's very sluggish this morning. All right, there we go. So now I'm sharing my screen now. So let me go. So let's see. And I'm going to move to papers. Can you see my screen?
Yep.
Okay, thank you. So I have a couple things I wanted to go over before the end of the meeting, and if you need to leave at the top of the hour, that's fine. The first one is this paper on volume segregation programming, and this is an early morphogenesis, new paper that came out in Physical Review E. So E is, I believe it's like materials, soft materials, and sort of biological stuff in physics. You know, it's Physical Review. They have like A through E, and then they have X, which is a open source journal, but or open access journal. This is E. So this is volume segregation programming and nematodes' early embryogenesis. And so nematode species are well known for their invariant cell lineage pattern during development. Combining knowledge about the fate specification induced by asymmetric division and the anti-correlation between cell cycle length and cell volume, we propose a minimal model to simulate lineage initiation by altering cell volume segregation ratio in each division, and quantifying the derived patterns, performance, and proliferation speed, fate diversity, and space robustness. This stereotypic pattern in C. elegans embryo is found to be one of the most optimal solutions, taking minimum time to achieve the cell number before gastrulation. So what they're doing here is they're taking the different founder cells, or the different, so what happens in C. elegans is you get to the 8-cell stage, so the cells divide, and you get what they call these founder cells. And the founder cells then start dividing, and they have they take up a certain volume of the embryo by virtue of them dividing. So you have your 8 cells, and then you have the descendants of those 8 cells, and they form a lineage. But then they take up space in the embryo. And so I think what they're doing in this paper is they're actually kind of simulating some of this and kind of figuring out what the optimal pattern is for this. And so when you go back, let's see what they have in here. So they have something called a logistic cleavage model. So they say that each lineage pattern is specified by a set of cleavage timings on the genealogical tree. So what they're doing is they're taking a cell, and we did a little bit of this in a couple of our papers. I think the one that we published in 2016, where we had the C. elegans data set and the Ascidian data set, and we were looking at the cell division timing. This is something where they're taking cell, like what they call a lineage tree of different cells. So the cells divide and differentiate along this lineage tree, and they're playing around with the timing. So they have the sequence of cell cleavage events during early embryogenesis. These timings are illustrated by solid circles. So each of these circles is a cleavage or a division, and this is over time. And they're not showing the tree, but you can imagine that at this circle, this single line branches into 2 lines. At this circle, the 2 lines, you might have an asymmetric division, where one of the lines up here divides into 2, and then this one continues until this circle, where then that divides into 2, and then here you have a 4-cell embryo that undergoes 3 divisions. It could be symmetric or asymmetric. So this is a lineage tree here, and you can see this in red, in blue, and in gray you have, and in green you have these, and actually in purple. So you have these different, subtrees or sublineages. So this is the AB sublineage. This is the P sublineage, which is the set of germ cells. This is EMS, C, D, and so forth. And so what they're trying to do is they're trying to, first of all, figure out from a set of founder cells, what the optimal division pattern is that fills up this embryo volume. So this is the embryo volume. If you fill it up with different sublineages, there's some optimality there that they're trying to capture. So they use this technique where they just look at these cell cleavage events on a one-dimensional time representation of time, and then they can measure it using this parameter. So A, which is or window of size S, which is this window of events, and then you have this A, which is the interval between events. So you basically, for any S, you want to have, A can be long or it can be short. You can have multiple A's, you can have one A, and so forth. So this is basically what they found. So based on our experimental cell trajectory analysis, we set S to 1.5 minutes and A to 3 minutes. In the following, we shall use cleavage clustering to classify and compare lineage patterns. So they do this for a number of different lineages, and they come up with these results here, where there's fate diversity and proliferation speed. So they're looking at, like, what kind of fates do you get, or how many divisions do you get versus the speed of proliferation. So do they proliferate fast? If they proliferate fast, is there a lot of diversity? Do you see this across sublineages? Do you see this, or is it just proliferating all in the same sublineage? So I don't really, I haven't dug really deeply into this paper, but basically the idea is that they're doing these simulations of different lineages, that they're claiming that these different lineage trees are solving this problem in different ways, and then they can find an optimal tree. So they're looking at a tree like this, where you have different divisions that are all symmetrical, and they divide at the same point in time, and they compare that with something where you get some asymmetric divisions, and then further asymmetric divisions, and so they're giving their results. They also find speed optimization. They find the results for that. They also have some spatial robustness. So there's a section on space robustness. So the continuous addition of new cells following sequential cleavages could jeopardize the canonical cell movement during mechanical equilibration inside the egg, leading to defect patterns. So some of these things where you have, some of the things where cells are dividing and finding their place in space. So you just keep adding cells. This can jeopardize sort of the mechanical equilibration and lead to defects in the embryo. So C. elegans solves this problem by synchronizing cell divisions in a temporally clustered set of events. So C. elegans has this clustered set of events that solves this problem that they're posing. To enforce this property among lineage patterns generated from a model, we introduced two time constants. This is where they put S and A in place. So these parameters actually represent a biological problem. And we did some simulations on this with respect to zebrafish embryos. Not this very problem, but we did some simulations of cell divisions to see if they were regular or if they had a different distribution from a random or normal distribution. And we found different results, that the biological embryo deviates from that control, which would be like a uniform set of divisions. So that's interesting. I didn't know that I guess this paper just came out. So there is actually a mechanical reason why embryos deviate from the normal distribution there. And so then they just have a little bit more simulation work here. The fast proliferating solutions of a relatively small number of distinct lineages. Within each lineage, and they mean sublineage, cell divisions are synchronized, hence only a few temporal clusters of cleavage events are needed to reach the target cell number n. On the other hand, for a solution with many lineages or equivalently a large f, which is another parameter, downstream temporal clustering and spacing of cleavage events become more intertwined under the minimal separation requirement. We named this phenomenon lineage interference. So this means that your lineage reaches a maximum number. So as they sort of, these two sublineages unfold, or multiple sublineages unfold in the same embryo, they interfere with each other, and so they can't proliferate uncontrollably. They have sort of an upper bound on how they can proliferate. Beyond this cell number, the solution space for lineage optimization shrinks rapidly. So when you get this, if you conform to this sort of lineage interference pattern, then any number of any cells in a single sublineages lineages are limited, and then beyond the cell number, you get you can't optimize for these different properties anymore. So this is an interesting paper. I didn't know. It just ties into a lot of the work that we've done in different papers, and I think it's really good work. again, I haven't really delved into this paper a lot, but this really applies to eutelic organisms mainly. So like C. elegans, eutelic is where this organism has the same number of cells in the adult. So in C. elegans, you have 959, and then a few more in the male. In the hermaphrodite, you have 959, and in the male, you have 1,024, I think, or something like that. But there's always, from individual to individual within that species, they always have the same number of cells, unless it's a mutant of some type. So this is what they mean by eutelic, and so that means that they develop to a certain number of cells, and then it stops, like the process stops. So if you're looking at like a mammalian embryo, this wouldn't necessarily apply, because the divisions would continue. There would be continual divisions. You'd have a lot of cell death. You'd have a lot of cells that can take on different fates, and then you have regeneration, which you don't really see in C. elegans too much. So this is only really something you would observe in a eutelic C. elegans-like organism. Nevertheless, I think it's good work. So that's that paper. Then I wanted to just kind of briefly touch on something to help us revisit this in our minds. This is the stuff on Cambrian embryos and development. So I know I've talked a lot about like the early embryos, like early meaning like geologically back in the Cambrian or Precambrian era, and the Dashanto assemblage of fossils in China has yielded us one of the earliest embryos, and this is an embryo that is something from just after the Great Oxygenation Event, when you're starting to get a lot of proliferation of different, orders and kingdoms of life. And so you get these early embryos. You start to see these clusters of cells, and you start to see this developmental process. And I think we've done a couple of papers on this where we've shown this, sort of these earliest embryos and what they look like. They don't necessarily look like the embryos of today, but you can definitely see they have properties of differentiation, and they have these properties of, cell, like colonies. They're not really cell colonies, but they're groups of cells that are developing into something else as an adult. And so you can start to see this lifespan start to unfold. So this is the first paper I found from this week. So they have this, this is the paper on current understanding of the Cambrian explosion, questions and answers. So this is just a very general review. In this paper, they're talking about sort of the Cambrian explosion and how that period, in the Cambrian period, is sort of where we start to see a lot of animal diversity and plant diversity. Actually, they focus on animal body plans here. And why this is relevant to embryos is because if you can imagine the early embryos being this very generic embryo that we can maybe recognize some features of differentiation and life history or stages of lifespan in. The Cambrian explosion then means that these embryos had to sort of incorporate these different body plans that were emerging at the time. And so during the Cambrian, you start to see all these different new types of organisms emerge. And not only do you see different types of organisms, but you have different body plans. And what that means is that you have different ways of having a body. So, you might have, say, like radial symmetry emerge, or bilateral symmetry, which just means that there are two sides of the organism that are similar, that they're sort of like not copies of each other, but they're symmetrical, meaning that there's a corresponding set of cells on the left side and the right side. Or you could have radial symmetry, where you have maybe six-fold symmetry. So you have 6 parts of the organism that are similar. So you have like a north, a south, an east, and a west, and then maybe like 2 other axes that have like similar organs or cell types or whatever. So that's what they mean by body plan. And this is an old idea going back to the German embryologists who proposed this. But the implication for embryos is that those body plans and all the things that come along with it had to be in these early embryos, or these early embryos had to sort of develop into these body plans. And so, this is something that this is just kind of a review of some of the diversity that was going on there, talking about molecular evolution, talking about some of the other types of things that explain the Cambrian explosion. So this is just a review of that period. This other paper that I have, a second paper, is a little bit more developmentally oriented because it takes this idea of the Cambrian explosion and it looks at it maybe a cause or a contributing factor. And it's so this question is, did homeobox gene duplications contribute to the Cambrian explosion? Why do we care about homeobox gene duplications? As I think in a previous meeting I talked about how homeobox genes are these Hox genes that are very important in development. And so, Hox genes are well known for their sort of being clustered on a genome in the same sort of location, and they kind of contribute to the different segments of an embryo. So you might have like an anterior and a Hox gene, or a middle segment Hox gene, or a Hox gene at the posterior end of the animal. And so they all map to that. They give it like sort of an orientation in space in the adult. And so homeobox gene duplication just simply means that these homeobox genes diversified and duplicated. So now you have more segments, you have more opportunities for new types of segments. And if you think about what was happening during the Cambrian explosion, you had this period where you had a lot of different body plans emerge. So if you have gene duplications in this homeobox region, then you have a lot of different possibilities for new phenotypes. And if you get rid of some of those genes in different lineages, then you can have radically different body plans. So this is where this paper kind of goes. They talk about bilaterian animals and some of the main features, that they have an anterior mouth, they possess muscle blocks for crawling and burrowing in sediments, they typically have an efficient through gut. Again, these are bilaterians in the Cambrians. We're bilaterians, but these are distant ancestors. So there's a through gut with a separate mouth and anus, so it's basically the digestive system. And then, of course, there are reasons why bilaterians develop this way. And so it builds on this molecular explanation of these homeobox genes that provide a means for body patterning, and then that led to some of these traits to emerge in the phenotype. So this is a nice paper that's sort of less speculative than, maybe more speculative than we'd like, but is actually pretty interesting. Then this final paper is called, it's by James Valentine, who has a Douglas Irwin, who are famous paleontologists. This is on fossils, molecules, and embryos, new perspectives on the Cambrian explosion. So they kind of bring everything together here. So this is what we talked about in the last paper, the Hox genes. In the paper before, we talked about the general tenor of the Cambrian explosion, how it led to these new body types. And in this paper, they're talking about sort of this, they're bringing it all together. They're saying that, we're looking at the fossil evidence, we're looking at what happened to different phyla, how they diversified. All living phyla may have originated by the end of the explosion. So every living thing on Earth, or at least animals, can be traced back to that time. There was nothing new after that emerged in terms of new phyla. So this was when a lot of that was established. And then molecular divergences among lineages leading to phyla record speciation events that have been earlier than the origins of new body plans. So the body plans actually maybe arose later. These new branches of life arose earlier. But then, so there's a molecular sort of precursor to these body plans. And then, of course, development is a way of mediating this into a phenotype and then into the adult. So they kind of talk about these the timing of the branches and how, the molecular mechanisms sort of predate the emergence of these phenotypes. And then, the timing of evolution of the developmental systems of living metazoans, which are animals, of these body plans is still uncertain. The distribution of Hox and other developmental control genes among metazoans indicates an extensive patterning system was in place prior to the Cambrian. So these patterning systems existed prior to the Cambrian, but they became diversified along with the lineages in the Cambrian. However, it is likely that much genomic repatterning occurred during the early Cambrian. So this means that these recombinations and duplications of Hox genes were occurring during the early Cambrian, involving both key control genes and regulators with their downstream cascades as novel body plans evolved. So that's all I have to say about that. I know that I think we've talked about some of these things before in terms of these sort of the origins of the embryo. And we wrote a paper on the origins of the embryo, but it was actually quite theoretical in nature. So this is like putting some real data to this concept, and so I think that's all I have for today in terms of papers. Did we have any comments on that?
I'm interested in the Hox genes, just from the standpoint of it changing the way the tissue flows.
Yeah. So that's one area. It's actually, the Hox genes are really kind of putting together these little modules. If you think about it in terms of a single Hox gene encodes a single module. And this is clearer in something like an insect or other, some like crustacean or something like that, where you actually see the segments pretty clearly in the embryo and then again in the adult phenotype. Our segments are less visible, but we have segments, and
Hox genes, yeah.
But I think I think we did a paper, and I can't remember. It was several weeks ago where they talked about this sort of. You see this sort of patterning in the sort of the physics of the embryo as a very early embryo, or I guess maybe it's a very basal embryo evolutionarily. So it's like something that's not, it's like it represents a very early organism, something that would have existed a long time ago, such as you can find them extant today. But they did some investigations into like the biophysics of it as well. So I think it's kind of interesting that those things are coming together. I'm going to have to bring together some of these readings and do a more formal list, because I think it's an interesting.
And the backbone, when the backbone develops in vertebrate, that's very interesting, and the Hox genes are involved as well as some interesting physics. So if one could put that together, it would be good.
Yeah. So I'm thinking more seriously about writing a paper on this, but it's like one of those things where I don't know, we have the expertise, although we don't have to be like paleontologists to do this. We can just simply say, this is what's been found, this is what we're interested in. We're kind of interested in like biophysics, and we'd like to know maybe how you get these early embryos and maybe what the biophysics were, what they were doing. Maybe it was like you had some embryo that was like free living, and then you added on like life stages afterwards. But that, like, I think in the paper that we wrote back in 2018, we kind of made some assumptions about like what was needed for an embryo to kind of form. There were like energetic constraints, and there were like strategic constraints by the cells, and so those are all like kind of in there. But it wasn't really grounded in any paleontology. I think I could imagine a paper where you had like a couple of hypotheses where you said, well, maybe we have this early embryo, and maybe we suggest that physical forces were important to sort of keeping these collection of cells together in a developmental stage, or maybe it was something else. You have these segments that emerge, and then how do they diversify? How do they not only in development, but how do you get new stages of life, life history after development? And why, I guess the question is why do you need development? And the answer maybe is just, you got to get from you got to reproduce. You don't want to, you can clone yourself. You could be a totipotent cell, have totipotent cells like the flatworm. But then, those are the only strategies? No, I mean, there are other strategies. You can lay eggs, and that's where you need an embryo, because you have a single cell that's then going to grow into the actually, we've seen the flatworm example, you have these totipotent cells, which are like eggs, and they just get kind of left in the environment. Well, in eggs, you need to have this protective layer. It kind of protects the single totipotent cells, and then they can become new organisms. And so a lot of those things,
I'm kind of interested in mammalian embryos and how they started, because there's a early stage there where there's very high pressure involved. So it's interesting.
Yeah. And then we can tie it into the oxygenation events, because we've noticed that there's this as soon as you get a certain amount of oxygen in the atmosphere, early in life you had very little oxygen. Then there were these two oxygenation events, and between those is this boring billion period where you don't see any diversification of life. And then you start to see it take off after you get a certain amount of oxygen in the environment. But we don't really know anything about pressure, because a lot of life at the time was in the sea. So,
going back to mammalian embryos, we start off with CO2 in our atmosphere, to develop. Like if you take a human embryo or a mouse embryo out of the mother, you have to keep it in a heated, high CO2 environment for it to live.
Yeah.
So that's interesting too.
Yeah. All right. Well, that's good. All right, Karan, did you have anything to say before we go? I didn't know if you had anything to say. Well, okay, so thanks for attending the meeting. And I think we'll have one more meeting before the end of the year. And then we'll be making a break for the holidays and then coming back in January, probably the first or second week. I don't know yet. But I'll send out an email.
So if I'm going to do the active matter lecture, it needs to be next week.
Yeah,
I'll make it a goal, because it is part of what I'm trying to write up right now. I have to write an 8,000-word essay.
Wow, okay.
So I'm sort of hoping to use part of my 6,000-word essay for it.
Yeah.
Not all of it. Anyway, yeah.
All right.
So,
well, that sounds good.
Okay.
Yeah, I'll put you down for next week, and then we'll see.
Okay.
All right.